Morning to everyone. Today I will present a study entitled Genetic and Molecular Mechanism of Thermo-Dormancy and Thermo-Inhibition in tomato seed. This study focused on how high temperature affect tomato seed germination and the genetic and molecular factor involved in heat tolerance. My name is Muhammad RIFAIE bin RAMLING from class M3 AT2282A. This presentation discusses research on the effect of heat stress on tomato seed germination and the mechanism that allow some tomato genotype to tolerate high temperature better than others. Seed germination is one of the most important stages in the plant life cycle because successful germination determines crop establishment and productivity. However, high temperature can negatively affect this process, with global warming causing more frequent heat stress events. Understanding how seed respond to high temperature has become increasingly important. There are two major responses of seed to heat stress. The first is thermo-inhibition, where germination temporarily stops but can resume when temperature becomes favorable. The second is thermo-dormancy, where prolonged heat exposure induces dormancy, preventing germination even when conditions improve. So our objective is to investigate how tomato seed respond to high temperature to identify genetic variation among different tomato genotypes, to examine the molecular mechanism regulating thermodormancy, to identify quantitative trait lossy or QTLS associated with heat tolerance and dormancy. This objective help researchers understand the biological basics of heat tolerance in tomato seed. So now we move to current issue. One major agriculture challenge today is the increasing global temperature caused by climate change, rising temperature, threatening seed germination and crop establishment. In tomato seed, exposure to high temperature can induce secondary dormancy, meaning the seed fail to germinate even under suitable condition later. Poor germination can lead to reduced seedling establishment, lower crop yield, and increased production costs for farmers. Therefore, development heat-tolerant varieties has become a priority in crop improvement program. So as we can see, this is the um, what do you call it? The map of I think the map of air temperature anomaly in January 2025. So the next slide is challenging the domestic status quo. Traditionally, scientists believe tomato seed dormancy was mainly caused by physical barriers such as the seed coat or endosperm. To test this idea, researchers removed the seed coat and endosperm from moneymaker tomato seed. Interestingly, germination still did not occur at 37 degrees Celsius. This finding showed that the problem was not caused by external seed structure. Instead, thermo-induced dormancy originated from physiological processes within the embryo itself. This discovery changed our understanding of how heat stress affects tomato seed. So, genetic variation versus P1. The researcher compared two tomato genotype. The first genotype was moneymaker or a cultivated tomato variety. seed completely failed to germinate at 37 degrees Celsius in both light and dark condition. The second genotype was PI, a wild tomato relative known as Solonu pimpilifolium (PI). Seeds show up to 80% germination under high temperature condition, particularly in darkness. This demonstrates significant genetic variation in heat tolerance. Another interesting finding was the role of light in wild PI seed. Light has an additional stress factor under high temperature, while in cultivated seed, light help break dormancy under normal temperature. So next slide is molecular and hormonal signaling. The molecular level several hormones and genes regulate. Thermo-Dormancy, the transcription factor FUS, CA3, FUS3 was strongly unregulated in Thermo-Dormancy and act as a master regulator. High temperature also increased the expression of ABA biosynthesis gene, specifically NCED1 and NCED9. Since ABA promote dormancy, higher ABA levels suppress germination. In contrast, genes involved in gibberellic acid production such as GA3OX1 and GA20OX1 were downregulated because gibberellic acid promotes germination. Its reduction further inhibits seed growth. Additionally, ethylene biosynthesis decreased under heat stress, contributing to reduced germination. Overall, heat stress shifts the hormonal balance toward dormancy rather than germination. So, result, which I will explain the graph. This figure compares the germination percentage of MM and PI seed at two temperatures, 25 Celsius and 37 Celsius, under both light and dark condition. At 25 Celsius, both MM and PI seed show high germination rate of around 90%, indicating that both genotypes perform well under optimal condition. However, at 37 Celsius, the responses were very different. MN seed show almost 0% germination, demonstrating extreme sensitivity to heat stress. In contrast, PI seed maintain approximately 80% to 90% germination, especially under dark condition, indicating strong thermotolerance. This result clearly show that Thermo-Dormancy is genotype dependent, the while PI genotype possess genetic traits that allow successful germination under high temperature, making it a valuable sources for breeding heat-tolerant tomato varieties. Main result point both genotype germinate well at 25%. 25 Celsius. MM failed to germinate at 37 Celsius. PI maintain high germination under heat stress. Heat tolerance is strongly influenced by genetic background. PI is a promising source of heat tolerance gene. Conclusion and future outlook. Last but not least, in conclusion, heat-induced secondary dormancy in tomato seed is highly dependent on genotype. This study demonstrates that while PI genotype possesses much greater heat tolerance than the cultivar MM genotype. Interestingly, researchers found that the major candidate genes such as NCED, GA-related gene, and FUS3 did not directly co-locate with the identified QTLs. This suggests that additional unknown genotype pathway may control the thermal tolerance. Future research should focus on fine mapping this QTL and using RNA sequencing to identify new genes involved in heat tolerance. Such discovery could support the development of climate resilient tomato varieties.